Bismillahirrahmanirrahim. My name is Aalia Abdul, the teacher of biology. Today we will discuss about the topic of mitosis. What is mitosis? What are the major events of mitosis? What are the phases of mitosis or why mitosis is important and fundamental for the living organism? All the answer of these questions have been covered in this topic. Before going towards the main topic, let's we have a brief introduction about the mitosis process. Wilder Fleming was the first scientist who discovered about the process of mitosis in living organism. Wilder Fleming observed that in a dividing cell, the cell nucleus passes through a series of changing called mitosis. What is mitosis? Mitosis is a process of cell division in which a cell divides into two daughter cells. Each cell has the same genetic material equivalent of the parent cell. Here some of the most important point related to mitosis process. Mitosis occur in a somatic cell. Mitosis process take place in eukaryotic cell. But in prokaryotic cell, the binary fission process take place. The mitosis process does not take place. Now, there are the two types of mitosis. One is open mitosis and second one is closed mitosis. In open mitosis, spindle fibers are formed inside the cytoplasm. This is called as open mitosis. Open mitosis process take place in animal cells. Now, closed mitosis, the spindle fiber forms inside the nucleus. This is called closed mitosis. Closed mitosis process occur in fungi and protista. Differences. What is the difference between somatic cells and germ cells? Somatic cells forming the body of organism. In somatic cell, the process of mitosis take place. In germ cells give rise to gametes. In germ cells, the process of meiosis take place. Now we will discuss about the events of mitosis. They are the two most important events of mitosis. One event is a karyokinesis and the second one is cytokinesis. In karyokinesis, the process in which division of nucleus take place. In cytokinesis, the process in which division of cytoplasm take place. When the cell completes its karyokinesis process, then cell completes its cytokinesis process. When both process is completed, then the process of mitosis would be considered as completed. Now we will discuss about the phases of mitosis. The first phase is prophase, second one is metaphase, third one is anaphase, and fourth one is telophase. Prophase. Prophase is one of the most important and major part of the mitosis. It covers 90% of the process of cell division. Genetic material in nucleus is thread-like, which is known as chromatin. At the start of prophase, chromatin convert into a condensed form called chromosome. Chromosomes attach with each other through centromere. Kinetochore protein is present near centromere. You can see from this diagram, colorful thread-like structure is present inside the nucleus. These loosened and thread-like structures are called as a chromatin. The colorful diagram is present to make the concept clear about the chromatin. Now, in the next diagram, there is a structure of chromosome. You can see there the four chromosomes are present. Two chromatids are attached with each other through a centromere. Near the centromere, there is a special protein is present which is known as a kinetochore protein. You can see at the both corner there is a microtubules are present. Microtubules are those proteins which are responsible for the formation of spindle fiber. Without the spindle fiber, the process of mitosis does not take place. These whole events are known as mitotic apparatus. Two pair of centriole give rise to centromere. But in a highly vacuolated plant, the cell nucleus migrate into a center before division. Now, there is a second phase of mitosis which is known as the metaphase. Spindle fiber in the metaphase grow up to a sufficient length. After reaching the sufficient length, kinetochore fiber attached to the kinetochore points. Centromere of the chromosome arrange themselves. When all these three events take place in metaphase phase, the whole event is known as the metaphase plate. You can see from this diagram all the chromosomes are aligned themselves on a straight line which is known as equator. When the chromosome arrange themselves on equator plate, this is known as the metaphase plate. Now, the third phase is anaphase. Chromosome moves in opposite direction. The force which is responsible for the movement of chromatid in opposite directions known as the pulling force. At the end of the anaphase, cell has succeeded in separated identical copies of genetic material into two groups. The process of anaphase can be seen through the diagram. You can see from this diagram, kinetochore protein is present, spindle fibers are present, and chromosomes are present. You can see from this diagram, both chromosomes means chromatid moving towards the opposite direction with the help of pulling force. The chromatid move towards the opposite poles. Now, this is the last phase of mitosis process. The telophase is totally the reversal of prophase. All the events which are taking place in prophase does not take place in telophase. Let's suppose we have nuclear membrane appear in telophase, but in prophase nuclear membrane disappear. In telophase, chromosome again uncoiled and converted into a thread-like structure which is known as a chromatin. But in prophase, the chromatid coiled in condensed form which is known as the chromosome. This is the structure of a telophase. You can see from this that there is a thread-like structure is present which is known as the chromatin inside the nucleus, and the last stage of mitosis which is known as telophase. After completing the phase of telophase, the process of karyokinesis is completed here. Now we will move towards the cytokinesis process. Now move towards the cytokinesis. There are the cytokinesis process is different in animal and in plant cell. In animal cell, cytokinesis occur by a process known as cleavage. In this process, the ring of microfilaments form. This ring is responsible for pinching the parent cell into two cells. In plant cell, vesicle form from Golgi apparatus, membrane-bounded disc called the phragmoplast. In plant cell, phragmoplast disc is responsible, dividing the cytoplasm into two equal parts. You can make the concept clear about the difference of cytokinesis process in plant and animal cell. You can see in a plant cell there is a Golgi apparatus are present. These Golgi apparatus form a body known as the phragmoplast. Phragmoplasts are responsible converted the one cell into two cell. But in the case of animal cell, the cytokinesis process is simple. In animal cell, the simple cleavage process take place. In a cleavage process, the cell making a forest and converted the one parent cell into two equal daughter cell. This is the whole description of mitosis process, and all the steps are in a brief discussed. First phase of the mitosis, the prophase, you can see. In this phase, centrioles are present, chromosomes are present, and centromere are present. In prophase, thread-like structure of chromatin converted into a condensed structure known as a chromosome. When the prophase process is completed, then the cell moves towards the other phase. You can see in the metaphase, All the chromosomes are arranged themselves on a straight line. The line is known as the equator line. When the all chromosomes arrange themselves on an equator plate, this whole event is known as the metaphase. The metaphase is the second phase of the mitosis. After the completion of second phase, the cell enter into a third phase. The third phase is anaphase. In third phase, the pulling force is responsible for moving the chromosomes towards the opposite direction. You can see from the third diagram, the two chromosomes are moving towards the opposite direction. Half chromosome are present at the one corner and half chromosomes are present on the other corner. Now, this is the last stage of the telophase. Telophase is totally the reversal phase of prophase. The events which are taking place in prophase does not take place in a telophase, but actually the reverse processes take place in telophase. In telophase, the chromosome loosen and form a thread-like structure which is known as the chromatin. Nuclear membrane around the nucleus will be reappear. After completing these four steps, the process of mitosis has been completed, in which one parent cell is divided into two equal daughter cells with the same genetic material. Now move towards some important short questions. There are the important short questions are: what is mitosis? What are the events of mitosis? What are the phases of mitosis? What is difference between chromatin and chromosome? What is difference between centromere and kinetochore? What is meant by mitosis apparatus? What is metaphase plate? What is telophase? Or what is the cytokinesis process? Jazakallahu khaira.